Imagine two people of the same age, say 50 years old, taking the same test that measures biological age. One gets a result of 42, the other gets 63. They stand side by side, they look about the same, neither one complains of any health issues. But underneath, at the cellular level, these are two completely different organisms. That is how biological age works. And the most interesting part of this story is not how to measure it. The most interesting part is what happens once you learn your own result and why so many people make the exact same mistake afterward. But let us take this in order. Chronological age simply counts the years since you were born. It is the number on your passport, and there is nothing you can do to change it. Biological age is something else entirely. It shows what those years have actually done to your body at the molecular level. And unlike the passport number, this figure is partly in your own hands. Take two men as an example. One is 55, a former marathon runner who still runs 3 times a week, does not smoke, and rarely drinks. When he took the biological age test, his result came back at 47. His colleague, the same age, who spent the last 20 years mostly at a desk, grabbing meals on the run and chronically underslept, got a result of 61. A difference of 14 years between two people born in the same year. Or take another example, a more personal one. A 48-year-old woman who had gone through a divorce and 2 years of intense chronic stress decided to take the test out of curiosity. The result upset her, 56. But instead of giving up, she treated it as a signal. A year and a half later, after fixing her sleep, moving regularly, and lowering her stress levels, she took the test again. The new result landed 5 years closer to her actual age. This shows the main point. The number is not a verdict. It is a starting point. How did scientists learn to measure this? The main tool here is called epigenetic clocks. It sounds complicated, but the idea is simple. It is an algorithm that looks at specific chemical marks on your DNA, marks that build up as you age, and uses them to estimate how old you are biologically. Not by your passport, but by the facts written into your cells. Across large groups of people, these clocks work remarkably well. Scientists have used them to figure out which habits speed up aging and which ones slow it down, and these findings are backed by solid research. But at the level of one individual person, meaning you, things are far less clear-cut. Different companies use different versions of these clocks. Some measure not accumulated age, but the current pace of aging right now. Others look at the process from a slightly different angle. And this is where things get genuinely interesting. Scientists recently compared 14 different versions of these clocks across 174 health outcomes. The results surprised even the researchers themselves. Not a single version came out as consistently superior to the rest. This means that if you take a biological age test with one company and then another, you could easily end up with 2 different numbers. And both could be technically correct, just measured by different yardsticks. What about telomeres? You have probably heard about telomeres too, the protective end caps of chromosomes that shorten with age. They often get mentioned alongside epigenetic clocks, and this is a common source of confusion. In reality, these are 2 separate tools measuring 2 separate processes. What is more, a recent study found that telomere length does have a causal link to healthy lifespan, while epigenetic clocks of aging did not show that same causal signal. Mixing these 2 tests together and drawing one combined conclusion is not quite accurate. There is another detail worth knowing, one that test advertisements rarely mention. Telomere length results from different labs usually cannot be compared directly. A lab can correctly tell you that your telomeres are shorter than your neighbours, but the exact number will differ from lab to lab simply because each one uses different calibration standards. What tests exist today? If after reading all this you find yourself wanting to try one of these tests, here are the main methods currently in use. Horvath's epigenetic clock, one of the first and most well-known algorithms built on DNA methylation analysis. It became the foundation for many commercial products. TruAge, a commercial test based on epigenetic clocks, one of the most recognized names on the market. It works on the same principle of methylation analysis. DunedinPACE measures not accumulated age, but the current pace of aging, useful for anyone who wants to know whether their aging is speeding up or slowing down right now. DunedinPACE, an earlier version of the same approach, also estimating the speed of biological aging through methylation patterns. CausAge, a newer model. Unlike the earlier ones, it focuses not simply on correlation with age, but on the changes that causally drive aging. Telomere length analysis, a method separate from epigenetic clocks, measuring the length of the protective ends of chromosomes. It reflects cellular replicative potential, but it is not directly comparable to results from epigenetic tests. Nucleosome positioning clocks, a newer and less widespread method that has only recently appeared, analysing the positioning of nucleosomes in cell-free DNA. Histone modification clocks, the newest class of tests, only beginning to reach the market. One important note applies to every method listed here. As mentioned earlier, different tests can produce different numbers for the same person, and none of them has yet been proven reliably accurate at the individual level. If you decide to take one of these tests, treat the result as a general reference point, not as a medical diagnosis. One scientific fact that changes the whole picture. Researchers at Stanford, led by Tony Weiss-Corey, discovered something that overturns the entire idea of a single biological age. It turns out you do not have one biological age. You have 11, one for each major organ system. The researchers analysed blood samples from thousands of people and found that each organ produces its own unique proteins, and the concentration of those proteins can be used to calculate that organ's own separate biological age. The results surprised even the researchers themselves. In roughly 1 out of every 5 people, one organ was aging noticeably faster than the rest, while the person felt completely healthy and had no idea anything was different. Some people, it was the heart aging ahead of schedule, in others the kidneys, in others the brain, and this had no correlation with how quickly the rest of that same person's organs were aging. The brain stands out in particular. According to the same research group, people with a biologically older brain died sooner, regardless of the age of their other organs. The scientists called the brain the true gatekeeper of longevity. What this means is that the single number most tests hand you is really an averaged picture, one that can hide something far more important, namely which specific organ actually needs your attention right now. Now to the part that matters most. So you took the test, you got a number, and this is exactly where most people make a mistake. They either panic at the result and give up, or they celebrate a good result and relax. Both reactions are wrong, because the real value of this whole story is not in the number itself. It is in the fact that this number can be influenced. A large study published quite recently painted a very clear picture. Smoking, excess weight, elevated blood sugar, and high blood pressure all accelerate biological aging. Physical activity and healthy eating, on the other hand, slow it down. And this is not some vague piece of advice along the lines of stay healthy. Another study, which combined data from 13 separate research papers, found that people with accelerated biological aging had strokes consistently more often. In other words, the number your epigenetic clock produces is not tied to some abstract corner of science. It is tied to a very concrete risk to your life. So what should you do with your test result? If you decide to take one of these tests, approach the result calmly. It is not a diagnosis and it is not a sentence. It is an estimate built on a particular algorithm, one that carries its own margin of error and its own version of the truth. What is actually worth paying attention to is not the number itself, but which direction it moves based on the habits you control every single day, whether you smoke or not, whether you move or sit still, what is on your plate. These 4 things influence your biological age more powerfully than any genetic test could ever predict. The number on the screen after your test is just a starting point. The direction it moves from there depends not on the lab, but on you. And if you would like to learn more about your own health and well-being, subscribe to our channel and join our community.